Morning. In this video, we are going to start by the functions of the lipids we have just studied in the previous video inside our human body. Of course, the first function is energy storage. And when we say energy storage, this is because 1 gram of lipids can give you 9 calories of energy if it is completely burned inside a calorie meter. And this is twice the amount of energy that is given by about twice the amount of energy that is given by 1 gram of carbohydrates or 1 gram of proteins. So lipids have the highest energy content per gram. But it's worth noting that in the human body, carbohydrates are first used to produce energy, meaning that the first foodstuff that is used to produce energy in the human body is the carbohydrate stuff, like, for example, glucose molecules. And then if all the glucose in the blood, or mainly glucose, it is the most abundantly consumed and produced and utilized carbohydrate inside our human body. If we finish all the glucose in blood, we start converting our glycogen stores in the liver into blood glucose. And when all this is depleted, we refer to fats. And when all fats are depleted for a reason or another, the body starts to break down proteins, which is a serious condition because you're actually breaking down your muscles and your enzymes, which are very important for normal human body functioning. So fats are essentially there to store energy just in case you don't have the first or the prime source of energy, which is glucose. But there is another function for lipids, which is the structural function, because some lipids can be bonded to a phosphate group to form a phospholipid, as we have seen in the structure of the cell membrane. We've just said that the cell membrane is a phospholipid bilayer. So this is the first aspect of the structural function of lipids. It is that we have the phospholipids that are derivatives of lipids that consist of a phosphate head and 2 hydrophobic lipid tail, and that form the phospholipid bilayer of the cell membrane. On the other hand, another lipid, which is cholesterol, which is a steroid, serves as an important component of the plasma membrane of the animal cells. So as we said before, cholesterol is not only a chemical molecule in the body that is harmful by depositing in the inner wall of the arteries and causing rigidity and sclerosis of the arteries, on top of which we might have blood clots and cardiac attacks or brain strokes. But cholesterol has another important useful aspect in the human body because it is important in the structure of the cell membrane. Now, the cell membrane, as we know, consists of a hydrophilic phosphate head and a hydrophobic lipid tail. So the phosphate head is hydrophilic because it is negatively charged, whereas the tails are hydrophobic. And if we have another phospholipid molecule, it's exactly the same. So the importance of the cholesterol molecule, if we look at it again, is that the cholesterol nucleus has the central steroid nucleus that is formed of 4 fused rings, and it has a hydroxyl group here, which is a polar functional group because the H might be lost in solution, the proton, to leave the oxygen as a negatively charged oxygen atom. Whereas on the other hand, we have here this hydrophobic side chain of cholesterol. So the cholesterol molecule that is present, actually we have several cholesterol molecules that are embedded in between the phospholipid molecules. So the hydrophilic part, you can imagine that the cholesterol molecule has a hydrophilic part that is charged and that will align with the phosphate heads or the hydrophilic parts of the molecule, whereas the hydrophobic part of cholesterol will form hydrophobic interactions with the hydrophobic tails of the phospholipid molecule. So by having done this, the cholesterol molecule will fix the phospholipid molecules in place and will help them, diminish the fluidity of the cell membrane. So it changes the fluidity of the cell membrane. It diminishes it. It diminishes or controls or regulates water penetration through the cell membrane, and it affects other properties of the cell membrane like compressibility, for example, without entering into too many details. But lipids also have an endocrine function, because we have just said that many famous examples of lipid molecules are steroid hormones, like the sex hormones, for example, that regulate the signs of puberty and help determination of sex or the gender. And at the same time, we have the glucocorticoids and the mineralocorticoids, glucocorticoids and mineralocorticoids. Now, the phospholipids, as we've said before, are considered modified lipids. Why? Because actually we have the backbone of glycerol that consists of 3 carbon atoms, and attached to the backbone of to the carbon skeleton of glycerol, we have 3 ester bonds that are related to 3 fatty acids. Fatty acid 1, fatty acid 2, and fatty acid 3. If we remove one of the fatty acids and replace it by phosphoric acid, then we will have here a phosphate group, and the molecule will be a phospholipid. As we said before, the phospholipids consist of a triglyceride molecule where 2 fatty acids are attached to the glycerol backbone, whereas we have only 1 phosphate radical. The 2 fatty acids that are attached to the glycerol backbone, this is the glycerol backbone. Of course, I've just written fatty acid 2, fatty acid 3, but actually they are long hydrocarbon chains like this, and therefore these lipid tails are nothing by the two lipid tails are nothing but the 2nd and 3rd fatty acids that are attached by ester bonds to the glycerol backbone. The 3rd hydroxyl group of glycerol is no more attached to a fatty acid through an ester bond, but it is attached to a phosphate group. That's why it is called a phospholipid, and that phosphate group is charged, and therefore it is hydrophilic, and therefore it attracts water, because polar likes polar and nonpolar likes nonpolar. And we said before that if we have molecules of phospholipids that are thrown in water or dispersed irregularly in water, they will again self-assemble and rearrange themselves into a double-layered structure called a bilayer. And just a reminder that in that bilayer, the phosphate heads that are hydrophilic or water lovers, if we have a container with water, will always be towards the water, whereas the lipid tails will always try to join each other and clump together to be away from water. So they will always arrange themselves in this bilayer arrangement, but not in any kind of bilayer, in a kind of bilayer that makes the phosphate heads always outer. And when we say outward or outer, we mean that they are towards water. We mean that they are towards water. So these ones are outer, and those ones are also considered outer. The phospholipid bilayer is very important in the structure of the plasma membrane because, as you know, it regulates the passage of substances inward and outward, and therefore it forms a boundary between the internal compartment of the cell and the external environment of the cell. We have studied this slide before, so we are not gonna repeat the explanation. And now we move on to a new group of organic materials, which are proteins. As we said before, proteins, just like carbohydrates, are macromolecules, or they are polymers, and they are therefore large molecules that are formed of small repeated monomers. And the monomers of proteins are the amino acids. So actually, we will see that several amino acid monomers can bind together to form a polymer, and that polymer will be called a polypeptide chain. A polypeptide chain. Poly means many, and peptide is the name of the bond between each two neighboring amino acids. So since we will have, for example, 4 amino acids linking together through 3 peptide bonds, then it is a polypeptide chain. So the monomer is an amino acid, the polymer is the polypeptide, but not most of the proteins are not made of a single polypeptide chain. That is usually linear and non-branched. And this is a major difference between the hydrocarbon and the polypeptide. Hydrocarbon can be branched, whereas the polypeptide cannot be branched. We always describe proteins as being complex molecules because usually you have this polypeptide chain that will fold, and this folded structure will be called the secondary structure, and then the secondary structure will further fold in a particular manner to give you a 3D configuration, in which case we call it the tertiary structure. And up to that stage, primary, secondary, tertiary, we only have one polypeptide chain. If the polypeptide chain is linear, then it is the primary structure. If the same polypeptide chain becomes simply folded like this, it giving you that helix-like structure, or sometimes a pleated structure like this. This again is a secondary structure. It's just simple folding of the molecule. But if starting from that helical structure or helix-like structure, or starting by that pleated structure, which is folded in a particular way, you further fold the molecule in order to give a globular, spherical, or 3D structure, then you are no more in the secondary, but in the tertiary structure, and we'll be discussing this in detail later on. Proteins, of course, have several important functions in the body. They are important, they help, mainly they are structural or functional, of course. Functional is very easy to understand because, as we said before, insulin, for example, is a peptide hormone that controls blood glucose level. Adrenaline and noradrenaline are polypeptide hormones that also control blood glucose level and other important functions of the body. They almost regulate every function in the body. But proteins are not only hormones, proteins are also enzymes. And enzymes, as you know, are involved in all biochemical reactions in the body. So without enzymes, or if only a single enzyme is deficient in the human body, this would greatly affect the function of certain, would greatly affect certain functions in the body. And like, for example, in favism, or or fava bean anemia, when you have a certain enzyme, which is glucose 6 dehydrogenase enzyme. When it is missing, it results in a rupture of the red blood corpuscles and a famous type of anemia that is related to the fava beans. And it is a serious type of anemia that happens when a child that has this enzyme being deficient eats, for example, beans or soya beans, for example, or eats burgers that contain soya beans. So needless to say that enzymes are important for life. Proteins, functional proteins, can either be hormones or enzymes, or they can be proteins that help the movement of muscles, like actin and myosin, that are responsible, as we will see in Unit 2 about the cell, they are responsible for contraction and relaxation of the muscle. But actually, I consider actin and myosin not only functional proteins, but essentially structural proteins. For example, in skeletal muscles, they are responsible for the striated structure of the skeletal muscle. So they are first structural proteins because they give the muscles, the voluntary muscles, a particular structure, and they are also functional proteins because they slide over each other. And when they slide over each other, the muscle will shorten or contract. Proteins, as we will see later on, are important for cell-to-cell signaling, meaning that if we have cell number 1 that wants to communicate with cell number 2, not through a hormone, not through an enzyme, then it might secrete particular molecules that we call signaling molecules or ligands. And these signaling molecules, if they find the specific receptor on the cell membrane of cell 2, these signaling molecules can bind with the receptor. And when they bind with the receptor, this is, of course, the signaling molecule or the ligand. And here we have the receptor, which is also a protein on an integral part of the cell membrane. So this is a receptor. And upon binding of the ligand with the receptor, this induces chemical changes inside cell 2. And cell 2, these chemical changes would be manifested as a particular change or a particular response in the cell. Of course, as we've said before, proteins are not here to give you energy. They do not store a big amount of energy. 1 gram of proteins releases about 4 calories of energy when burnt in a calorie meter. And of course, you all know the sources of proteins, whether they are animal proteins like fish, like meat, like poultry, or vegetarian proteins, plant proteins like in beans and lentils and all the leguminous plants and in peanuts. How are peptides synthesized? As we've said before, amino acids bind together to form peptide bonds. But how does an amino acid look like? The amino acid, any amino acid, consists of a central carbon atom that has 4 single bonds. The first single bond is with a hydrogen atom. The second bond is with a carboxylic group, and that carboxylic group might lose the proton in an aqueous solution and leave the oxygen atom with a negative charge. and on the other side, we have the amino acid NH2 at a particular pH. That proton that have been lost by the carboxylic group might be gained by the amino group, which will then be converted into ammonia, into an ammonium group. But up till now, we will look at the amino acid in its simplest form without focusing on the changes that occur in a solution. The fourth group is an R group that is considered a variable group. This R group is called the side chain, and this R group will differ from one amino acid to another and will determine many properties of the amino acids, because that R group can be a hydrophilic group or a hydrophobic group. It can be an acidic group or a basic group. Actually, we have one acidic group, which is the carboxylic group in the amino acid molecule. And why is it considered an acidic group? Because it can lose a proton in a solution. So it gives protons, so it is considered acidic. On the other hand, we have an amino group, and that amino group is considered a basic group. So the carboxylic group is always considered an acidic group in solutions and in aqueous solutions, and the amino group is considered basic. Why? Because it can accept a proton. And if it accepts a proton, then it will be, or it will carry a positive charge, and it would be NH3 instead of NH2. Because most amino acids in nature have one carboxylic group that is acidic and one amino group that is basic. So most amino acids are considered to be acidic. Most amino acids are considered to be neutral, neither acidic nor alkaline, because they have one acidic and one basic group. But if it happens that the R group is acidic, for example, another COOH group, then this amino acid will have two carboxylic and one amino group, and it would be acidic. Or on the other hand, if that R radical is another amino group, then the amino acid as a whole will be considered basic, because it would have two amino groups and one carboxylic group. In nature, we have only 20 different amino acids that can unite to form polypeptide chains. And because every polypeptide chain will depend upon, or the characteristics of a polypeptide chain will depend upon the number of those amino acids, the order or the arrangement of those amino acids in the chain, as well as the type of those amino acids, then we can have thousands of possibilities of polypeptide chains that might form hundreds of thousands of protein molecules. Again, at a neutral pH, the carboxylic group is an acidic group. Why? It's written in green at the bottom of the slide, because it can release a proton and carry a negative charge or acquire a negative charge. And on the other hand, the NH2 group or the amino group is at a neutral pH basic, because it can gain a proton and acquire a positive charge. Why are we studying the structure of amino acids? Because they are the building blocks of polypeptide chains, and they can form together, as we said before, peptide bonds, and hence the name polypeptide. For example, here we have amino acid number 1 and amino acid number 2 that bonded together to form a short chain that is made of essentially these two amino acids with a peptide bond in between, and that's why it is called a dipeptide. So a dipeptide is actually formed of two amino acids, and in between we have one peptide bond. How is the peptide bond formed, and how does it look like? just like any bonds that form upon having building up reactions, then the peptide bond is formed by removal of one molecule of water. So you take H from the amino group of one amino acid, and you take OH from the amino acid from the other amino acid. You remove both of them. You remove this water from the medium of the reaction. Therefore, again, we are taking off water, so it is a dehydration synthesis reaction. And as we said before, when forming disaccharides, the dehydration synthesis reaction has another name, which is the condensation reaction. And this is how a peptide bond is formed, by the removal of one molecule of water. Of course, after having obtained this dipeptide, if you want to break it down again, then you would hydrolyze it. So on one side, we have a condensation reaction. And if we want to reverse the condensation reaction, we carry out a hydrolysis reaction. As you can see, condensation reactions build up larger molecules from smaller monomers. So condensation reactions are considered building up reactions or anabolic reactions, whereas on the other hand, hydrolysis reactions allow you to get two simple molecules from an original larger molecules, and therefore we can consider hydrolysis reactions catabolic reactions or breakdown reactions. The last item to discuss on that slide is how the peptide bond looks like. Now, after having removed this hydrogen and that hydroxyl, we end up by having this nitrogen atom having a bond here and a bond there, but missing a third bond. Similarly, after having removed the hydroxyl here, you end up by having a carbon atom that has only three bonds, or three shared pairs of electrons, two with the oxygen atom and one with the nitrogen atom. So the carbon atom also requires to form a bond. So nitrogen and carbon end up by forming bonds.